The overall goal of this experiment is to make a specialized microfluidic device and use it to measure the lifespan and cellular phenotypes of a single yeast cell.
This method can help answer some of the key questions in the yeast aging field, such as why mother cells age and die, and what are the earlier molecular events that drive the loss of cellular homeostasis and eventually cell aging and death. The major advantage of this technique is that we can track mother cells, track their lifespan, and various molecular markers at the same time, so we can look for molecular events that correlate with lifespan.
Though this method can provide insight into yeast aging, it can also be applied to other areas, such as monitoring cellular responses to perturbation that require cell tracking for many generations.
Generally, individuals new to this method will struggle because microfluidic device fabrication, cell loading, microscope setup require a lot of practice to perform successfully. Visual demonstration of this method is critical, as it is difficult to properly load the microfluidic device with yeast.
To begin, fabricate a silicon wafer mold for the device using standard techniques as described in the accompanying text protocol. Place it feature side up into a 15 cm Petri dish. Secure the mold on the Petri dish with tapes. Next, place a clean weighing boat on a balance and tare the balance. Pour 50 grams of PDMS base into the weigh boat, and then add 5 grams of PDMS curing agent. Stir the PDMS base and curing agent with a disposable pipe.